Good morning, students. Today we are going to discuss about crustacean acid metabolism in bryophyllum plant species. In these plants, the dark reaction is somewhat different from C3 as well as C4 plant species. How does starch is prepared within these xerophytic plants, example bryophyllum, aloe vera, like Opentia cactus? In these plant species have a somewhat different anatomical structure. That's what is see here. First of all, we can try to understand the stomatas. Stomatas are open during night time and closed during daytime. They open during night time because of to reduce the loss of water. Because of plants are growing in the less amount of water, scarcity of water area. If they open during daytime also, greater amount of water have been lost through these stomatas by the process of transpiration. Hence, the plants have its own adaptation, that is the stomatas are closed during daytime, open during night time. So they are closed during the daytime. But here, during daytime, sunlight is available, then water molecules undergoes, then chlorophyll molecules are used for this light reaction. But under the light reaction, ATP, NADPH form of energy is produced. But during daytime, carbon dioxide is not available. So, but during daytime, light reaction is going on. But during night time, there is a scope of carbon dioxide availability. That's why during daytime they have a chance to go for light reaction as well as dark reaction, because of during daytime whatever that during night time carbon dioxide is taken, that carbon dioxide is stored in the form of malic acid. That malic acid undergoes to some changes to release the carbon dioxide to do the dark reaction during the daytime. How does it? You can see this now. Now during night time they open the stomatas, carbon dioxide comes inside. This carbon dioxide, bicarbonate ions form is there. They can react with phosphoenol pyruvate, hence converts into acetate. In the sense, phosphoenol pyruvate have the 3 carbons. They combine with the carbon dioxide. Altogether, this converts into 4 carbon compound oxalo acetate in the presence of phosphoenol pyruvate carboxylase enzyme. The oxalate is converts into malate by the help of enzyme is called as malate dehydrogenase. By consumption of NADH, the NADH is converted to NAD+. The malate is converted into malic acid by losing 2 protons. The malic acid is stored within the vacuole structure. This process is going on during the only night time. This process we can call as an acidification. In the sense, vacuoles have become acid. During daytime, the process here, whatever the malic acid is stored within the vacuole, during daytime there is a light reaction going on. ATP, NADPHs are available. Hence, the carbon dioxide want to release, but here there is no chance to get the carbon dioxide from the atmosphere. But this carbon dioxide is wanted. This carbon dioxide is released from the malic acid wherever they store in the vacuole structure. Now, during daytime, malic acid is converted to malate. The malate is converted to oxaloacetic acid. The oxaloacetic acid is converted to pyruvate as well as carbon dioxide. This carbon dioxide is undergoes to Calvin cycle as usual dark reaction. But pyruvate is undergoes to starch formation. This process takes place with the chloroplast structure. Whatever the starch is stored here, in the sense pyruvate is converted into starch with the help of enzyme is called as phosphoenol pyruvate dehydrogenase enzyme. This starch during night time they stores with the chloroplast, but starch is converted into triose phosphate. Triose phosphate is converted into phosphoenol pyruvate. Within this, during the night time, in the sense, starch is a source of phosphoenol pyruvate. Then here malic acid is source of carbon dioxide. So carbon dioxide is released during the daytime, undergoes to Calvin cycle. At the end of this Calvin cycle, starch is prepared. Means dark reaction in these bryophyllum plants takes place with the help of day and night during timings as well as with the help of chloroplast and vacuole. Whatever that reaction is going on from the malic acid is converted into pyruvate is called as an deacidification. The process which is going on from phosphoenol pyruvate into the oxaloacetic acid to the malic acid is called as an acidification. Dear students, now we can try to understand the photorespiration process. The photorespiration also called as an C2 path or glycolate path. This process was discovered by Otto Warburg. It is also called as an Warburg effect. The Warburg effect in the sense, in the plant species, photorespiration process is a common, photosynthesis process also common. But when the carbon dioxide is available, photosynthesis process takes place. When the oxygen is available, respiration process takes place. If the concentration of carbon dioxide is less, then the concentration of oxygen is more. Instead of photosynthesis, there is a chance for respiration. The respiration takes place with the help of sunlight. Means in the process of dark reaction, respiration takes is called as an it is also called as an photorespiration. Means when the photorespiration takes place, the rate of photosynthesis will be affected. This effect is called as an Warburg effect. Now we can see that photorespiration process in the chloroplast. In the chloroplast, RUBP, then already present in the chloroplast, RUBP can accept, means RUBP react with oxygen to form as 2 moles of phosphoglyceric acid as well as 2 moles of phosphoglycolate. Phosphoglycolate consists of 2 carbon in each one, then phosphoglyceric acid consists of 3 carbon in each one. This conversion is possible in the presence of Rubisco enzyme. Rubisco enzyme have the multiple functions. One is they can react with, they can accept the carbon dioxide by RUBP, and also they can accept the oxygen by RUBP. This photorespiration takes place in some extreme conditions. Instead of carbon dioxide, if oxygen concentration is more, then carbon dioxide concentration is less. That carbon dioxide oxygen is accepted by chloroplast, it is undergoes to series of reactions. The respiration takes place in the presence of oxygen, but that is in the chloroplast, is called as a photorespiration. In this one, RUBP is converts into 2 phosphoglyceric acid plus 2 moles of phosphoglyceric acid plus 2 moles of glycolate. In this one, 3 carbons are present, here 2 carbons are present. Now second one is an phosphoglycolate undergoes to dephosphorylation process in the presence of phosphorylase enzyme. Then it is converts into 2 glycolate, then each one consists of 2 carbons. Now this 2 carbon glycolate is transfers from chloroplast to peroxisomes. These 2 glycolates undergoes to, then 2 moles of glycolates converts into 2 glyoxylate plus 2 moles of hydrogen peroxide. This conversion is possible by addition of 2 moles of oxygen. Glycolic oxidase enzyme is used. Where does oxygen comes? Here hydrogen peroxide is splitting into water plus oxygen. This oxygen is going to use along with glycolate to convert into 2 glyoxylate. Means water molecules plus oxygen converts into hydrogen peroxide. Hydrogen peroxide is converts into oxygen plus water molecule. So oxygen will be added along with this glycolate, and then peroxisomes, hydrogen peroxide is released from glyoxylate within that peroxisomes. Now glyoxylate is converts into glycine by transaminase enzyme, then by addition of some amino acids. Then glycine consists of 2 carbon of, here 2 carbons are present, it is also 2 moles of glycine is forms. Means glycolate, each one consists of 2 carbon, then 2 moles of glycolate converts into 2 moles of glyoxylate, then 2 moles of glyoxylate, each mole of glyoxylate consists of 2 carbons, again they converts into 2 moles of glycine, each one consists of 2 carbons. Now this glycine is transfers to this mitochondria, then mitochondria have that third organelle here, then 2 moles of glycine, each one consists of 2 carbon, going to lose 1 mole of ammonia, along with that then 1 mole of carbon dioxide. When the 1 mole of carbon dioxide is going to lose here, then 1 carbon dioxide is reduced, then it became that serine, then 3 carbon compound of 1 mole of serine is forms. Here is 2 moles of glycines, each one consists of 2 carbon, 2 twos of 4 carbons are present, but 1 carbon is loosed in the form of carbon dioxide. The remaining third carbon compound is formed, that is an serine. In this conversion, NAD is became NADH, energy formation is possible. This conversion is called as an decarboxylation. This is one of the enzyme is used called as an decarboxylase enzyme. At last, at end of this mitochondrium, then 1 mole of serine is formed. This serine is transfers to this one peroxisome, then in the form of serine. Then here as a 3 carbons are present, 1 mole is present. This serine is converts, means transfers, converts into hydroxypyruvate in the presence of deamination process. Deaminase enzyme is used, and here amino acids have been emitting from this one serine. It also consists of 3 carbon compound, then 1 mole of hydroxypyruvate is forms. The 1 mole of hydroxypyruvate is converts into glycerate, then by the enzyme is called as a reductase enzyme. It is also possible that NADH utilization takes place. NADH is become NAD plus, means some amount of protons are added here. Glycerate. The glycerate is transfers from peroxisome into the chloroplast. The glycerate consists of 3 carbon each one. The 1 mole of glycerate is present. Then the glycerate is going to convert into phosphoglyceric acid. Then by utilization of ATP is became ADP. When inorganic phosphate is added to the glycerate, it became phosphoglyceric acid. This process called as in reduction. This takes place in the presence of reductase enzyme. Then phosphoglyceric acid, 3 carbon compound, going to recycle as formation of RUBP. In the sense, 2 RUBP molecules, then 2 oxygen molecules, 2 moles of phosphoglyceric acid, 2 moles of phosphoglycolate. This is continuous as a series of reactions. In this process, the importance of photorespiration is dark reaction is going to take place then by in the presence of carbon dioxide, but the carbon dioxide is not available within the chloroplast, but carbon dioxide is emitting in the mitochondrial structure. This carbon dioxide is going out of this mitochondria and it is a transfers to the chloroplast. Then the dark reaction is undergoes to Kelvin cycle, but chloroplast itself do not have the capacity for absorption of carbon dioxide to do the dark reaction, but chloroplast absorb the excess amount of oxygen and that oxygen converts into carbon dioxide. This carbon dioxide is going to use in the dark reaction. This is the specialty of this photorespiration process. Now we can try to understand that factors affects the rate of photosynthesis. One is an factors are here, 2 types are given here as an external factors and internal factors. The external factors are classified into 4 types. One is an availability of sunlight and availability of temperature, availability of water and carbon dioxide concentration. Come to the external factors, there is a 2 factors are there. One is leaf as well as protoplasmic conditions. Then external factors, the first one availability of sunlight. The light is not enough of. In that light there is a 3 important factors are there: light intensity, light quality, light duration. What is meant by intensity? Then light which is coming as a VIBGR. So then wavelength, different types of wavelength lights have been emitting from sun star, but all the wavelength lights are not suitable for photosynthesis. Only the VIBGR light is suitable for photosynthesis. How much light is emitting from sun star and how much captured by leaf is one of the factor. Then quality of the light. Out of this VIBGR, all the wavelength lights are not suitable. Here red light is useful for maximum photosynthesis, then blue also useful for rate of photosynthesis process, but green is used for minimum level of the photosynthesis. Then duration, that leaf, in the leaf chloroplast, chlorophyll is present. When a plant exposed to sunlight continuously, the rate of photosynthesis is measured. That when the leaf and chloroplast is exposed to sunlight discontinuously, the rate of photosynthesis is estimated. But when the light is falls discontinuous, the rate of photosynthesis is maximum. In the sense, in the duration, discontinuous light gives more rate of photosynthesis than that continuous light falls. Then come to the temperature. When the temperature level have been increases, the rate of photosynthesis also have been increased up to certain level. Above certain level, the rate of the availability of temperature is increases, even the rate of photosynthesis falls down. Now water condition, as availability of water, the rate of photosynthesis have been increased. Concentration of this carbon dioxide. When the carbon dioxide concentration increases from 0.03 to 0.04, 0.5, 0.05, the level of photosynthesis, the rate of photosynthesis also have been increased. Even the concentration is more than 0.05, like 0.05, the level of photosynthesis, the rate of photosynthesis falls down. In the sense, carbon dioxide is also one of these factor effects on the rate of photosynthesis. Come to the internal factors, leaf anatomy. In the leaf anatomy, the amount of mesophyll cells, the amount of chlorophyll, the amount of chloroplast also impact on the rate of photosynthesis. The number of leaf, the more number of leaf is present, the rate of photosynthesis also will be more. The leaf number is less, the rate of photosynthesis will be less. Size of this leaf. If the size is very broader, the rate of photosynthesis will be maximum. If it is small size, photosynthesis is least minimized. Age of this leaf, young leaf have the more photosynthesis capacity. When the leaf age how many increases, the rate of photosynthesis is falls down. Then protoplasmic factors, hydration means the amount of protons available in this chloroplast impact on the rate of photosynthesis and also enzymes activities. If the enzymes are more active, the rate of photosynthesis will be more and more. These are the factors are there, external as well as internal. Two types of factors are present. This is a law of limiting factors. The definition was given by the Blackmans. The definition is given like it's: if a process is conditioned as to its rapidity by a number of separate factors, the rate of the process is limited by the factors that is present in a relative minimal value. Does that mean if something process is affected by more than two factors, but some of the factors are acting as a limiting factors? What does that mean? We can see now. This is then factors affecting the photosynthesis process is given as a graphical. These are two factors are impact on this photosynthesis process. One is a light intensity, the second one carbon dioxide concentration. That typically you can see here. At this point, the light intensity is minimum, the carbon dioxide concentration also minimum. When the light intensity availability, when the light intensity have been increased, the rate of photosynthesis also have been increased. When the minimum means maximum level of light intensity is given here. As per the concentration, as per the light intensity is increased, the level of carbon dioxide concentration also have been increased. Then level of carbon dioxide has have been increased, the level of light intensity available to have been increased, automatically the rate of photosynthesis also have been increased. When that light intensity is maximum, the rate of the availability of carbon dioxide also maximum, then the rate of photosynthesis is maximum. If the light intensity available to have been increased, if the carbon dioxide availability is not increased up to the light intensity, the rate of photosynthesis does not increases as per the light intensity increases. The rate of photosynthesis will be constant. In the sense, even the light intensity increases, the carbon dioxide concentration increases, it's okay. The rate of photosynthesis have been increased. If the carbon dioxide concentration is not increased, even though light intensity increases, the rate of photosynthesis will be does not increases, it is false as a constant. This is a graph explains the relation between the light intensity and the level of carbon dioxide, the concentration of carbon dioxide. If the condition is a plant in the leaf, chlorophyll, chloroplast is present, the level of the light intensity have been increased, then availability of carbon dioxide have been increased, the rate of photosynthesis also have been increased. When the light intensity is less, the rate of photosynthesis also minimum. When the light intensity is increased, the rate of photosynthesis is maximum. The availability of carbon dioxide also maximum. The condition is here, light intensity have been increased, but the level of carbon dioxide does not increase. Then the rate of photosynthesis will be constant. In between the light intensity and carbon dioxide availability, the carbon dioxide is acting as a limiting factor. And this second graph explains as the relation between the light intensity and carbon dioxide. Here at this point of 0, a point of A, the light intensity is minimum and the availability of carbon dioxide is minimum. The rate of photosynthesis is 0. When the light intensity have been increased, the level of carbon dioxide have been increased, then that rate of photosynthesis have been increased from A to C. When the light intensity is increased, the availability of carbon dioxide is not increased, then the rate of photosynthesis will be constant, that is in C to D. Then the second condition is the rate of the light intensity is increased and also the rate of photosynthesis means availability of carbon dioxide also increased, the rate of photosynthesis is increased. Up to certain level, the light intensity is increased, but the level of carbon dioxide is not increased up to the light intensity works effectively. Then the level of photosynthesis will be constant. Then the level, the second one is then, third one is then, the level of intensity is increased and also availability of carbon dioxide also increased. Then the level of photosynthesis also increased. then here maximum. Whenever the level of photosynthesis increased, then the light intensity again increased. The level of carbon dioxide, availability of carbon dioxide is not increased, then the rate of photosynthesis will be constant. In the sense, wherever the light intensity is increased, according to the light intensity, the level of carbon dioxide increases, the rate of photosynthesis also will be increased. If carbon dioxide is not increased up to the level of light intensity, the photosynthesis rate will be constant. When the carbon dioxide started to increase up to the light intensity, the level of photosynthesis will be raised up. Again, that if the carbon dioxide is not sufficient for light availability, then as per the light availability, the rate of photosynthesis will be constant. Whenever the carbon dioxide as well as light intensity is increased continuously, the rate of photosynthesis have been increased. In the sense, then carbon dioxide is one of this limiting factor compared with the light intensity. In the sense, out of many more factors, one factor sometimes will be affecting as a limiting factor. All the factors are available, but one factor is not available up to the maximum, then the rate of life process will be affected. This is called as a limiting factor. From previous questions, from the today discussed topic, we can see some few. First question is, a plant in your garden avoids photorespiratory losses, has improved water use efficiency, shows high rate of photosynthesis at high temperature, and has improved efficiency of nitrogen utilization. In which of these following physiology groups would you assign these plants? Means in these plant species, they live at high temperature, the loss of the photorespiration does not take place, and that they have the maximum photosynthesis process at high temperature. Cam plants, crustacean, acid metabolism. Option 2 is a nitrogen fixer. Third one is a C3 plants. Fourth one is a C4 plants. Answer is in C4 plants do not have the photorespiration. They can live at high temperature. They have the maximum respiration process without photorespiration process. Now we can see the question number 2. The correct sequence of the cell organelles during the photorespiration is. Option A is given as a chloroplast, Golgi bodies, mitochondria. Given 2 option is in chloroplast, rough endoplasmic reticulum, and dictyosomes. Option 3 is given as a chloroplast, mitochondria, peroxisomes. Option 4 is given that chloroplast, vacuoles, and peroxisomes. So then given option is, and third one is the right one, that is an chloroplast, peroxisomes, mitochondria. Actually sequencing, but these 3 organelles are used in that photorespiration. The third question is, cam plants, crustacean acid metabolism helps the plants in conserving water, secondary growth, disease resistancy, and reproduction. So cam plants conserve their water because of during night time they have the open the stomatas only for carbon dioxide intake, but during daytime they do not open the stomatas. During night time they can open the stomatas. We can see the fourth question. Cranch anatomy is one of these characteristic feature of the leaves of cranch anatomy. We were discussed last. In the C4 plant species, there is a cranch anatomy is present. In the tropical plant species, the photosynthesis dark reaction takes place in the 2 mesophyll as well as cranch anatomy. Example for example, potato, wheat, sugarcane as well as mustard. The example for cranch anatomy is in sugarcane as well as maize. Then fifth question is, during photorespiration the oxygen consuming reactions occurs in. Then option 1 is given as a stroma of chloroplast, option 2 is given the stroma of chloroplast and mitochondria, option 3 is given the stroma of chloroplast and peroxisomes, option 4 is a gran of chloroplast and peroxisomes. So then here photorespiration takes place, oxygen is consuming in the chloroplast as well as peroxisomes. My dear students, we have completed the photosynthesis in higher plants by these 5 parts of the videos. Next video we are going to discuss about respiration in the plants.